So thank you, Sherry, for this very, nice talk and the introduction and inviting me here. So yeah, indeed, I've been asked to talk about biogenesis, and I'm going to try to say a bit more about the heterogeneity, as we heard already. So can I? Oops, sorry, no. I'm not sure there is a pointer, but. No, okay. So yeah, so this is a slide where we, in my group, we have represented heterogeneity. So as you see here, you have on the right, on the right-hand side, you have the vesicles forming from the plasma membrane. This is an image of a eukaryotic cell releasing vesicles, and you have. Okay, thanks. So the plasma membrane-derived vesicles on the right. Then you have these vesicles that form first inside internal compartments that contain intraluminal vesicles, the multivesicular bodies. And when these multivesicular bodies fuse with the plasma membrane, their intraluminal vesicles are released outside, and that's what's called exosomes. What this slide illustrates also is that the size of exosomes is constrained, is limited by what we observe as the size of intraluminal vesicles of multivesicular bodies. And it's generally considered as between 50 and 150 nanometers, more or less. But this size can be also the size of the smallest vesicles that bud off the plasma membrane, which in fact we like to call now ectosomes rather than microvesicles, because the term microvesicles has been used for a little bit of everything. So the term small EV somehow would cover the EVs that are smaller than a given size, and generally we say 150 nanometer, and that covers both exosomes and the small ectosomes. Whereas the large EVs would be the EVs that are larger than, let's say, 150 nanometers, if you can measure that. And these large EVs have to come from the plasma membrane, because you don't see such large EVs inside multivesicular bodies. And the last thing illustrated in this slide is that the cells also do not release only EVs. Of course, we in the EV field have been really focused on that, but somehow we forgot that the cells also release lots of other things that are not vesicles, as Sherry already mentioned, and including these particles that have been described in 2018. That's why we put them in this short review. The exomeres, now there are more of them, the supermere, etc. And also, of course, there are the soluble proteins that are released by the cells. So in terms of heterogeneity of EVs, so this is another illustration of the same diversity. So different groups in the world focus or want to focus more on the particular type of EVs. For instance, the exosomes, the ones that form in multivesicular bodies, they are mainly, and they are studied a lot by these groups, Guillaume Vanil, Michael Pertel. I'm not going to quote them all. I'm just going to show that many groups are working on many different EVs. Then you have other groups that work mostly on EVs that form from particular extensions of the plasma membrane, like cilia, nanotube, filopodia, etc. There are also lots of groups that work mostly on the large EVs. And since we had initially drawn this scheme, actually there have been much more large EVs, very large EVs described, like the migrasomes or the exopheres containing parts of mitochondria. So there is really like expansion of our knowledge or what we recognize as heterogeneity of the EVs. And now just a little comment on the non-eukaryotic EVs that Sherry already very nicely introduced. So of course, you have bacteria. The point I want to make here is that bacteria do not have internal compartments, so they cannot make exosomes, right? So you cannot say that bacteria make exosomes. They make extracellular vesicles of different types. There was a nice illustration in Sherry's talks. Here I've just put two other illustrations of outer membrane vesicles from these two slides. And actually the gram-positive bacteria apparently are also able to release EVs which have to cross the cell wall. So it's still a bit surprising, but still, I mean, they also make EVs. But again, these are not exosomes. And another aspect I just wanted to mention in this slide is that when you isolate EVs from tissues, from intact tissues, from eukaryotic generally tissues, you need to treat the tissue to separate the cells from each other and from the extracellular matrix. So you have to keep in mind that because of the, the extension of the plasma membrane that I showed in the previous slide, even when you do gentle enzymatic digestion, you can destroy some of the cells. So it's always a bit difficult to prove that what you have isolated from a tissue was initially really extracellular rather than intracellular. So just keep that in mind. So now, when you isolate EV, if you want to say that you isolate exosomes or ectosomes or a specific subtype of biogenetically secreted EV, the problem is that once they're outside the cell, the different EVs, they have really overlapping physical properties. So on the top here is illustrated the size of the EVs and the exosome, or the smallest ectosomes overlapping in size, but also overlapping with apoptotic EVs and apoptotic bodies and overlapping with retroviruses, which actually could be considered as an extracellular vesicle, and also overlapping with some lipoproteins. And at the bottom, it's the density when you float or you pellet these EVs in a gradient. And again, the different types of EVs, their range of densities overlap. So you don't have a very precise definition of an EV subtype based on its size or its density. And when you isolate EV, actually the techniques of isolation, so this is a figure from the last MyZev, showing the different techniques of isolation that are most commonly used and presented on a grid of recovery, efficiency of recovery, vertically versus specificity of EV isolation horizontally. So all these methods are differently distributed on this grid, but what's the point here is that the way these methods isolate EV is based on their physical properties and not on their biogenesis or subcellular origin. So when you isolate an EV, again, you cannot really know whether it was coming from a multivesicular body or from the plasma membrane or from any other unknown origin. So what can be done to try to identify the subcellular origin of EVs? So the only real way is actually to follow live in secreting cells the EV-associated molecules and to show that these EV-associated molecules come out of the cells and end up in structures that are outside the cell. So this has been done, for instance, by using some specific molecules that have been shown to be specifically expressed on particular subtypes of large EVs coming from the plasma membrane, the migrasomes, the cilia-derived EVs, or long plasma membrane protrusions. Using fluorescent forms of these molecules, these groups could show that these large EVs were formed and released from the plasma membrane of these cells. Concerning exosomes, there has been a very nice tool that has been generated by the group of Michael Perktel initially, but then used also by the group of Alisa Weaver and Guillaume Van Niel, etc., which is a CD63 molecule. So CD63 is one of the tetraspanines that's often used as a marker of EVs. I'm going to come back to that in the next slide. And a variant of this CD63 fused to a GFP that's sensitive to pH, the pH lurin. So the CD63 pH lurin is not fluorescent when it's inside an internal acidic compartment, but it becomes fluorescent when it's released outside where the pH is not acidic anymore. So this trick has been used by these groups to show that there was indeed some multivesicular body fusion with the plasma membrane and release outside of the vesicles bearing CD63. In my group, we have done another thing, and the question I was really not sure to understand for several years was, why would we use CD63, as I mentioned, the tetraspanin, but also CD9, another tetraspanin, as markers of exosomes? When you look at their distribution in cells, they are completely different. So you see here CD63 in red is inside the cell in internal compartments, where CD9 is really mainly at the cell surface. So why would CD9 be a marker of exosomes in this condition? So we tried to answer this question by engineering cells to express CD9 or CD63 in the system that's called a rush, which means retention using a selective hook, where you retain a secreted protein inside the endoplasmic reticulum by putting with a hook with a biotin, streptavidin tag. And when you put biotin, then you release the protein and it follows its intracellular trafficking. So we could follow this way the intracellular trafficking of both CD63 and CD9. And we could show that, see that it was that they were both following the same traffic initially, but then diverging. We also, in this paper, we also used a mutant of CD63, which had been described a very long time ago, actually, by the group of Paul Luzio, which is a mutant of the lysosome addressing motif, which thus remains at the plasma membrane. This mutant CD63YA remains at the plasma membrane or in very small compartments close to the plasma membrane. And when we expressed this mutant in the cells, what we could see was that the amount of the CD63 that was recovered in the smallest EV pelleted at high speed was higher when it was the mutant CD63 that was expressed than when it was the wild-type CD63, suggesting that actually more EVs in this particular cell line at least come from the plasma membrane than from the multivesicular bodies. And so the general conclusion of this work was that, in fact, CD63 and CD9, they travel together in the cells. They reach the plasma membrane, but then CD63 is very rapidly endocytosed, goes to MVBs. But still, while it's at the plasma membrane, it can end up in EVs budding from the plasma membrane. But CD9, since it's staying at the plasma membrane, is much more abundant on EVs coming from the plasma membrane than CD63. And conversely, CD63 is more abundant on EVs, on exosomes, on EVs coming from MVBs. But neither of them is really uniquely expressed on exosomes or on ectosomes. So now the other part I wanted to talk about in this talk was the technology or the techniques that we try to use to identify the subcellular origin of EVs. And the one way that's very commonly used is to try to block intracellular biogenesis machineries using drugs or siRNA or shRNA or guide RNAs, etc. So this has been done a lot. Using, for instance, to block all these molecules of these pathways that had been proposed to be involved specifically in exosome secretion. So the escort or the neutral sphingomyelinase that generates ceramide, or the synthenin or the phospholipase A2. So drugs or guide RNAs have been used to block all of these machineries, and this was resulting in decreased release of EVs that were interpreted as exosomes. But in fact, when people looked more carefully, there were also not only exosomes, but these drugs could also affect the budding of, in particular, escorts are involved in the budding of viruses, but also ectosomes from the plasma membrane. The ceramide is also, when you block neutral sphingomyelinase, you increase the secretion of large vesicles. So all these pathways are interconnected. And also one thing that has to be kept in mind is that when you block these pathways, you may also block the secretion of other regular secretory pathway aspects. So really, upon inhibition of an intracellular machinery, it's really important to keep in mind to ask oneself whether it's only one subtype of EV that's affected and whether other secretion pathways are affected. And we particularly in my group, we realized these issues in these 3 successive papers that we published. So the first one was that we could clearly show. I don't see my pointer. Yeah, we could show back a long time ago, back in 2010, that RAB27, which was known to be an intracellular RAB protein that's involved in secretion of secretory granules, so that RAB27 inhibition would decrease the secretion of exosomes. And these exosomes, we had defined them as CD63 and CD81 expressing vesicles. But when we tried to use this to block exosome secretion in vivo, then we realized that the cells, when we were blocking exosome secretion, we were also blocking secretion of some soluble proteins. So it was not only specific of exosomes. And then the last aspect that was published later by Matthias Ostrowski's group was that it was also blocking indirectly the budding of viruses from the plasma membrane by preventing bringing of PIP2 to the plasma membrane. So indirectly, blocking RAB27 was blocking also the secretion of ectosomes, I mean vesicles budding from the plasma membrane. So it's probably the same kind of issues that can happen in the other studies where people have used blocking RAB11 or RAB35, which are RAB mostly associated to recycling or early endosomes, to block secretion of EVs, or RAL-A or B that is mostly involved apparently in regulation of biogenesis of MVBs, but maybe also in other things. And for all these studies, really, I think it's important to ask whether they are specific of exosomes or they are doing other things to other EVs. So to summarize, there is no really means to specifically inhibit or promote the release of a given subtype of EVs, either exosomes or ectosomes, and any treatment will have eventually direct or indirect effects on other pathways. The difficulty in characterizing exosomes versus ectosomes is that there are no generally specific marker accepted for a given EV subtype. You can say that CD63 or MVB-enriched molecules like LAMP may be used as a proxies of exosome marker, but you must keep in mind that they all go through the plasma membrane at some point and thus may end up in EVs coming from the plasma membrane. And that it's not only late multivesicular bodies that are sources of exosomes, but also other internal compartments like early or recycling endosomes. And and again, for the ectosomes, the plasma membrane-derived EVs, there are maybe some more specific markers than what's used so far. CD9 is more enriched on ectosomes than on exosomes, and we have identified other molecules like CD98, which is possibly a proxy of ectosomes, but that needs to be verified in other cell types. Then another big question, I think, that remains here since the exomers or the non-NVEPs have been described is where do they come from? Where are they secreted from the secretory pathway? Are they really coming from the live cells, or are they byproducts of broken cells or from the medium? So that remains a big question. So in the slides, I was asked also to put a translational focus, if I could. So I just want to highlight here that I think it's important for a translational application to perform comprehensive studies of EV's heterogeneity to understand, and this is valid for any pathological or physiological system, because it's really important to determine which EVs will have the effect that you're really looking for, and because some EVs may have one effect, some other EVs may have the other effect, and if you use a mixture of the two EVs, you may not see any effect because you have a mixture of positively and negatively acting EVs. So in terms of rigor and reproducibility, the recommendations are that when you manipulate secreting cells to affect the secretion of a given subtype of EV, first of all, do not consider that results published by other groups on other cells or other models will apply to your own model without double checking that it's the case, because every cell behave differently. Second of all, you really have to keep in mind that you need to analyze the consequences of manipulating the cells on the other secretion and the other physiological functions on the cells, especially if you want to use that for in vivo studies to say that secretion of EVs in vivo do this or that. So you should really analyze the amount of or the composition of roughly separated EP subtypes, not only the 200K or 100K pellet where you think you have your exosomes. You must analyze the viability or the senescence of your cells, and you must analyze also an array of secreted products, and even modification of secretion of a single protein, a soluble protein, may in vivo have strong consequences. And finally, on the horizon, I hope that we are going to reach more knowledge on the biogenesis, the subcellular origin, and the functions of the non-vesicular EP. So of course, there is a lot on lipoproteins in the literature, which can be considered a type of non-vesicular EPs. But these other non-vesicular EPs, like the exomeres, again, where do they come from, where they are formed, this is really unclear to me. And to understand better the respective contributions of the EV and VEP soluble factors and the corona that's resulting from all this on EVs. And maybe eventually we are going to have to decide that we cannot really define EV subtype by their biogenesis pathway, because it's so difficult to define that when you analyze a bulk population of EVs, but rather by a surface protein that would be of interest for your function. So that's something that's maybe the evolution in the future. I don't know. So I'm done, and thank you for your attention.